All right, it's my pleasure to introduce Dr. Giovanna Zupičić. She's a research associate at the Croatian Veterinary Institute in Zagreb, Croatia. She works in the Laboratory for Aquatic Animal Diseases, which serves as the EU National Reference Laboratory for Fish, Crustacean, and Mollusk Diseases. She holds a PhD in molecular biosciences with a research focused on the diversity of Vibrio harvii in the Mediterranean aquaculture industry. Dr. Zupičić has been actively involved in several EU-funded research projects contributing to the advancements in aquatic animal health and sustainable fish farming. So take it away, Ivana.
Thank you, and welcome. Good afternoon, everybody. I will tell you one part of our research in our lab regarding to the bacteria Vibrio harvey from Mediterranean Sea. What bacteria Vibrio harvey? It's emerging disease agent in global aquaculture. It causes very serious losses during the summer months. As I check the references, I saw that also in the USA there are some troubles with this bacteria. The disease is called vibriosis, and it's caused several decades in a Mediterranean Sea, it causes big economical losses, mostly on most foreign Mediterranean fish as European sea bass and gilded sea bream. This is the opportunistic bacterium is associated with serious production losses in marine fish farm with the potential, it can potentially eliminate the whole population mostly of juvenile fish and has a big impact of the aquaculture industry. As you can see in this picture here, it doesn't need some bigger grow requirements. It grows on marine agar and TSA with added salt. The colonies are white to yellow. It grows at 22-24 Celsius degrees. It's a gram-negative, luminous, oxidase-positive, facultative anaerobe bacteria. It causes significant losses during the summer months. Here is the picture of sea bass. You can see the hemorrhages around the mouth, some changes around the eyes, on the opercula, on the hemorrhages on the fins, and also very specific for this bacterium, hemorrhages on the intestine. Primarily, this bacteria was described in Asia on shrimp farms. But now, due to climate changes in all the world, and so in the Europe and the Mediterranean region, we find very serious problems with this bacteria now. What we did in this research, we take the 16 strains of bacterium Vibrio harvey, isolated from European sea bass Tetrarchus labrax and gilded sea breams Parus aurata, from Mediterranean region, from 7 different countries, from Cyprus, France, Croatia, Italy, Spain, Tunisia, and Turkey. In this is here the map of Mediterranean regions, and in red dot you can see the spots where the fish were taken for this research. This was in a collaboration with other scientists in the Mediterranean region. They send their sample to us so we can do the research. When we get all the samples, we check them to be sure that this is the Vibrio harvey bacterium. We performed the PCR, which was based on the TOX-R gene, which is described by Pang et al. From 2006. It's a region which is very well conserved in this bacteria. And after performing the PCR, you have PCR products around 3082 base pairs, which shows that bacteria was amplified. After that, we in our lab in Croatia, we start the sequencing using the ONT MinION and Oxford Nanopore technologies for 16 strains. And 11 out 16 strain we send to outsource company for Illumina high-sec sequencing. Maybe you will ask you why we didn't send all the samples. This was a part of the project, and we don't have enough funding in this project for sending all the samples for Illumina sequencing. After we obtain our long reads from Menaion sequencing, we assembled the genome, and also for the samples for which we had short reads from the Illumina sequencing, we polished our genome from obtained by assembly by Menaion sequencing with short reads with Illumina, and we get better quality of or better coverage of the genome is in that way for the samples we have this sequencing. After we get done and assemble the genome, we search for the virulence genes in VFDB database, which is available online, and we search for antimicrobial resistance genes in AGI identifier, which is in the frame of CARD system, which is also available for research online. It's a big and good database. And we constructed a phylogenetic tree, which was based on TOX-R gene, as I mentioned before, which is well conserved for this bacteria species. Except this in silico analysis, we perform also in vitro susceptibility test using this diffusion method and microdilution test. This part of research was also part of our project with other labs during across the Europe. Different labs from Europe use the same method of this dilution and micro dilution test with their own samples to detect the cutoff values for these bacteria, as the CLCI, Clinical and Laboratory Standard Institute, doesn't provide the cutoff value for Vibrio harvae. In this way, we wanted to provide more information and guidelines for the future. For this test, we used 12 different antibiotics, and the microdilution plate, as you see here in the picture, was designed from Thermo Fisher Scientific, specifically with the concentration of antibiotics and antibiotics we wanted for this research. And the antibiotics we use in this research are one which are important for the aquaculture. After we perform all this research, we get some results. Here you can see that the whole genome of bacteria Vibrio harvae, it's around 6.2 kilobase pairs. It depends, it's variable in different strains. The bacteria has two chromosomes. The chromosomes are circular. One is slightly bigger than another, like one chromosome is 3.6 kilobase and another is around 2.2 kilobase pairs. And also we use the BUSCO tool, bioinformatic tool, to see which shows the completeness of genome. And when we polish the sequences obtained by Menaion sequencing with the Illumina sequencing, you see that in that strains, the BUSCO, the coverage and completeness of genome was very good, was 99 to 100%. And for the samples we didn't polish with Illumina sequences, the completeness was slightly less, like 65 to 78%. Also, we find plasmid in our genome. Usually, there was 1, 3 to 5 plasmid. The average size of the plasmid was 78 base pairs. This is the visualization of the chromosomes. This was the strain specifically from Croatia. You can see one chromosome slightly bigger than another, and the plasmid, and the regulatory elements with a similarity greater than 80% with the ATTC strain from the NCBI database are marked turquoise here. After searching for virulence genes, we find the total 171 virulence gene in 16 research strains. And out of these, 150 of genes were present in all isolates, all strains. This is 88%. In this table, you can see the virulence factors we find and the genes on the right side, which are coding for these virulence factors. These genes, which are marked here, CYT-A, C, B, D, which are important for iron transport, and the gene AC, which is important for heat stable cytotoxic enterotoxin, are found only in bacteria from the family Vibrio, which were present in the research database. And this is also the good point. Here you can see the heat map of these virulence genes. Here you can see the list of the genes. In red are the genes which are present, and the blue color marks the genes which are absent in the particular strain. And here on the right side are strains. We also searched for antimicrobial resistance genes. We found 5 different antimicrobial resistance genes. Here is the list. They are coding for different resistance of different antibiotics, and they have different resistant mechanism. And you can see that most of them were present in all our research strains, except the last 2, which were not present in all our research strains. In vivo results from the diffusion and microdilution tests, as I told you before, there is no CLCI standardized cutoff values for the bacterium Vibrio harvey. And there is no specific threshold we can talk about. So we are talking about the inhibition zone in millimeters. And we found that all resistant, those strains were resistant to ampicillin, which is expected because it's known from the other papers that Vibrio harvey is resistant to ampicillin, but we don't know the resistance mechanism. And one Croatian strain has a smaller zone of inhibition from gentamicin than other strains. There was a resistance to sulfametaxazole visible in 2 strains. The strain from Spain had a smaller zone of inhibition of sulfametaxazole than other strain. One strain from Turkey had smaller zone of inhibition for oxytetracycline. This all can indicate that some countries are using antibiotics, some of them are not using antibiotics, and all the information we obtained from this project we put in the paper. You can see here in the right side. So if you want, you can check about this data. And about the microdilution test, this is the graphical presentation of the results. This is the list of antibiotics, and these are concentration of antibiotics in the well. As I told you, the Vibrio harvey strains were resistant to ampicillin, so you can see that for all the concentration there was a resistance, and for example for oxytetracycline you can see that this is the resistance for these 2 concentration, but 14 samples were sensitive. We have the minimal concentration in resistant in this concentration. And also we did some phylogenetic analysis, and you can see two phylogenetic trees in both of them. You can see three different phylogen clusters. These are strain in red. You can see that all our research strains were grouped in the same phylogenetic group, which improved their homogeneity. And other strains are strains from all over the world, which are took from the NCBI database, which are available and open. And to conclude, there, after assembling the genomes of 16 strains, we determined that all strains share very similar composition of virulence genes regarding to the geographic origin. Some strains have the CIT genes and the AST genes, which were not available, or which were not, which other bacteria from the same family doesn't, from the research database doesn't have. And it was found that some strains exhibit a sensitivity to gentamicin, chloramphenicol, and oxytetracycline in vitro. And despite that, in their genome, there are genes for antimicrobial resistance. And these findings may suggest that the potential transfer of antimicrobial resistance genes by plasmid, which the bacteria can release very easily during the subcultivation, and this can be the reason why we get different results. And considering these all obtained results, along with the implementation of good production practice and the good biosecurity, which is most important on the fish farm, it's possible to develop unique regional guidelines for prevention and control of these diseases, which are causing lots of economical losses nowadays. And if you want to find more about this topic, we have published this paper, and you can read about this research. And thank you all for your attention. If you have some questions, you can ask me.
Great. Thank you, Ivana, for your presentation. That was very interesting to hear what's going on in the Mediterranean Sea. If we're behind schedule, so if people have questions, feel free to drop them into the chat, and perhaps, Ivana, you can answer them there, or they can follow up with you via email afterwards.
Okay. Thank you.
Thank you.